na Angel L. Pupi, a student from grade 7 St. Matthew. For today's video, I will teach you how to make your own edible animal cell. But before that, let's define what is an animal cell first. Animal cell. Animal cells are the basic unit of life in organisms of the kingdom Animalia. They are eukaryotic cells, meaning that they have a true nucleus and specialized structures called organelles that carry out different functions. The ingredients that I will be using are icing, gulaman with condensed milk, gulaman with sugar, Hansel crackers, gummy belts, Jamin milk, dotted candies, half and whole nips, flat tops, gummy worms, gummy beans, bar nuts, and chewing gum. First, put the gulaman with sugar on top of your base. This gulaman with sugar represents the cytoplasm of the cell. Cytoplasm. The cytoplasm is home to many activities of the cell as it contains molecules, enzymes that are crucial in the breakdown of the waste. The cytoplasm also assists in metabolic activities. Cytoplasm provides shape to the cell. It fills up the cells, thus enabling an organelles to remain in their position. Second, put the icing on the side of your cytoplasm and your base. This represents your plasma membrane. Cell or plasma membrane. The plasma membrane or the cell membrane provides protection for a cell. It also provides a fixed environment inside the cell. And that membrane has several different functions. One is to transport nutrients into the cell and also to transport toxic substances out of the cell. Third, put your Hansel crackers on the top of your cytoplasm. The Hansel crackers represents the nucleus. Step 4. On top of your nucleus, put the flat tops. The flat tops represents as your nucleolus. Nucleus. The nucleus consists of nuclear pore, chromatin or DNA, nuclear envelope, and nucleolus. Its function is to control and regulate the activities of the cell, such as growth and metabolism, and carries the genes, structures that contain the hereditary information. Nucleoli are small bodies often seen with the nucleus. Nucleolus is a region found within the cell nucleus that is concerned with producing and assembling the cell's ribosomes. Following assembly, ribosomes are transported to the cell cytoplasm where they serve as the sites of protein synthesis. Put the gummy worms beside the nucleus. The gummy worms represents as the smooth and rough endoplasmic reticulum, or simply called ER. Rough endoplasmic reticulum are simply called rough ER and smooth ER. Their function is to produce proteins for the rest of the cell to function. Step 6. On top of the cytoplasm, put the gummy belts. The gummy belts serves as the Golgi apparatus. Golgi apparatus or Golgi complex and Golgi body. The Golgi apparatus is a membrane-bound organelle found in most cells. It is responsible for packaging proteins into vesicles prior to secretion and therefore plays a key role in the secretory pathway. Step 7. Put one gummy worm on side of the base. This gummy worm serves as the flagellum. Animal cell. Animal cells are the basic unit of life in organisms of the kingdom Animalia. They are eukaryotic cells, meaning that they have a true nucleus and specialized structures called organelles that carry out different functions. Mitochondria. Mitochondria are membrane-bound cell organelles that generate most of the chemical energy needed to power the cell's biochemical reactions. Chemical energy produced by the mitochondria is stored in a small molecule called adenosine triphosphate or ATP. Step 9. Put the bar nuts on the vacant spaces. The bar nuts represent as the microtubules. Microtubules. They are involved in maintaining the structure of the cell and, together with microfilaments and intermediate filaments, they form the cytoskeleton. They also made up the internal structure of the cilia and flagella. Step 10. For centriole, I will use cutted gummy worms coated with cutted gummy belts and put it on the vacant part of the cytoplasm. Centrioles. Centrioles play a role in organizing microtubules that serve as the cell's skeletal system. They help determine the locations of the nucleus and other organelles. Step 11. Put 3 gummy beans on every vacant places of your cytoplasm. The gummy beans represents as the lysosome. Lysosomes. Lysosomes are involved with various cell processes. They break down excess or worn out cell parts. They may be used to destroy invading viruses and bacteria. Step 12. Place the half nips and the whole nips on the vacant places of your cytoplasm. The nips represents as your vesicle. Vesicles. Vesicles are the basic tool used by the cell for organizing cellular substances. Vesicles are involved in metabolism, transport, buoyancy control, and temporary storage of food and enzymes. They can also act as chemical reaction chambers. Step 13. Place the dotted candies as your ribosomes. Ribosome. Ribosomes have two main functions, such as decoding the message and the formation of peptide bonds. And lastly, step 14, place the Jamin milk on the vacant places of the cytoplasm. The Jamin milk represents your intermediate filaments. Intermediate filaments. The most important function of intermediate filaments is to provide mechanical support for the plasma membrane, where it comes into contact with other cells or with the extracellular matrix. Unlike microfilaments and microtubules, intermediate filaments do not participate in cell motility. You make your own edible animal cell. And the last part of this video is the fun part. It is tasting time! Tasty! And that's the end of my video. Thank you for watching and I hope you enjoy my video. Bye!